Good morning, everyone. This morning I'm going to be speaking about horseflies, and specifically the genus Hybomitra and a new species that we discovered. So horseflies are known vectors of pathogens. They're able to spread viruses, bacteria, and even protozoans. But unfortunately, there hasn't been a lot of research on the horseflies in the Afrotropics in the recent years. The most comprehensive work on the horseflies was done by Oldroyd in the 1950s. Pam Usher did some work in the 60s and 70s, but since then there really hasn't been a lot of research done. More recently there are a couple of papers that have come out, but unfortunately there are some very doubtful identifications, and so that's making a bit of a mess of the literature at the moment. The genus Hybomitra is actually considered to be holarctic, but there are 15 species that are listed in the catalogue of Afrotropical Diptera, and in the manual of the Afrotropical Diptera, Cheney actually suggested that Hybomitra should not in fact be an Afrotropical genus. It should in fact be lumped with Tabanus. The problem is that the genus Tabanus is at this stage a bit of a dumping ground, so everything that people don't know what to do, they just stick it into Tabanus. So that's making it more complicated. We went on a field trip in the Drakensberg region of South Africa, and we discovered a species which we believe does fit into the characteristics for Hybomitra. And the characteristics for Hybomitra are hairy eyes and an ocellar tubercle, and so very few characteristics that separate it from the genus Tabanus. So our field trip, we were working in the Natal Drakensberg region. That purple dot there is the area in which we collected the specimens, and this is specifically the Manx Cowl Nature Reserve. So there's some photographs there of the area in which we were doing our collecting work. The photograph on the right, you can see there's a little white triangle. That's one of our Manitoba traps, which we use for collecting horseflies. And if you have a closer look at what the Manitoba-style traps look like, they're basically a tripod. They've got 3 metal legs that are joined together at the top with a metal ring. And from that, you can suspend a hook, which you attach a black ball to, and it allows the ball to blow, sort of move around in the wind. And then it's covered by this plastic skirt, and there's a collecting head at the top. We use a chemical lure. We use something called octanol, which is basically designed to smell like animal. And then the black ball gets hot in the sun, so it radiates heat. And along with the smell of animal, we're basically trying to create a fake animal that the flies then see the heat and they smell and they are attracted, and they then attack the ball and they try to bite the ball. And they get trapped underneath that plastic skirt, and then they fly up and they get caught in the collecting head at the top. And these traps are quite effective when you have nice sunshine, because it's a thermal trap. You need the sun to heat the ball up. So the specimens that we collected, we took a hind leg of 4 of the specimens in order to do DNA extraction and sequencing. We also then downloaded all the available sequences from GenBank and BOLD for CO1, 16S, and 28S. And we analyzed sequences doing maximum likelihood analysis and also drew network analyses. We then tried to track down the descriptions of all the Afrotropical Hybomitra that are listed in the 1980 catalogue. And this is actually really challenging because a number of the publications are in very, very obscure journals. They were very difficult to come by. And a lot of them are in they're not in English, so we had to translate them. Some of them were in French, some of them were Portuguese. It was a lot of work to get all of the literature together. And the descriptions of some of the species are very vague. There's one, in fact, that isn't a complete description. It's a comparison to another species as to why it's a different species. So it was very difficult to actually get concrete descriptions for each of the different species. But we did our best with that. We came up with diagnoses for all the species so that we could at least have in one place diagnosis for each of the species of the Hybomitra from the Afrotropics. We then, having looked at all of the literature, we're convinced that our specimens that we had collected was in fact a new species, and so we did the description and we took photographs with those. We also then mapped the type localities so that we could show where they're actually from in Africa. So this is what our new species looks like, Hybomitra phalaris. It has the hairy eyes, it has the ocellar tubercle, which is why we believe it fits into the genus Hybomitra. And what was very interesting is that we caught another specimen, which we're referring to as CF saxicolus. There is a species that was Tabanidae saxicolus. We've actually moved to being Hybomitra saxicolus, based on the fact that it has the hairy eyes and the oscilloturbical. But this one falls, it's got very similar patterning, but it is not identical to the saxicolus. Unfortunately, we only caught one specimen, so we'd rather have more specimens to be 100% sure before we do the description of it. And we did sequence it as well, so we've put it into our analysis, and you'll see that it actually falls with the other Hybomitra. So the molecular analyses, the green bars are the whole Arctic Hybomitra, and the blue bar is the Afrotropical. There were some sequences in bold that came from South Africa, but they were from river assessments, so they were actually from the larvae. And they group with our Hybomitra. But unfortunately, because they were larvae, we don't have any identification species level. We just know that they are Hybomitra. So we don't know which species they're from. You can see the network diagram on the left, also just showing that the whole Arctic and the Afrotropical Hybomitra definitely separate into two distinctive clades. But you can see the whole Arctic Hybomitra are paraphyletic by the fact that there's a Tabanidae grouping that sits in the middle of them. And the Tabanidae lower down is also paraphyletic. So, as I said, Tabanidae has been a dumping ground where everyone's just thrown species they don't know what else to do with. So it really has a lot of work that needs to be done. We mapped the type localities, so you can have a look there at all the different species. There's definitely a Western Africa grouping, there's a Central African grouping, and then sort of Southern Africa. There are one or two exceptions. Cabanassi from Mozambique is actually no longer a Hybomitra. It has been synonymized with a species in the Tabanidae, but we've included it because it was in the 1980 catalogue. And you can see that we've got a couple of species in South Africa. Those ones that are in blue are the ones from bold that I was talking about that are just from the larvae. So we need to do a lot more collecting to actually see, to confirm which species they are. So what do we conclude from all of this? Well, the main characteristic is hairy eyes, and hairy eyes is a very subjective character because how hairy is hairy? There are actually some Tabanidae that have slightly hairy eyes. In fact, some of the descriptions for the Hybomitra say they have bare eyes or they have absolutely no description about the eyes. So it's a very subjective character and one that we don't think is very reliable. The problem is that the only other character is the oscilloturbical, and that only occurs in the females because the males have holoptic eyes, so you don't get the turbical. So if you don't have, you generally catch females, you don't often catch males, but if you catch a male, you can't be sure. So we really need to find new characters to describe the Hybomitra to separate them from Tabanidae. And we desperately need to find new, fresh material that we can actually do the molecular work on as well, because some of those holotypes, they don't even know actually where they are. So in the descriptions that we had, they list the locality of where it was collected, but some of them don't list at all where it was deposited. Some of the collection deposition institutions don't exist anymore because they were there for specific reasons and they no longer exist. There's no indication of where the material was sent. So yeah, we tried really hard to track some of them down, and there's just a big question mark. So it really is challenging, and it would be really good to get fresh materials so that we could actually do molecular work as well, so that we can link the taxonomy with the molecular work. And potentially we might actually be looking at needing to describe a new genus for the Afrotropical species. Thank you very much.
Thanks, Kirstin. I have a question. Did you figure or photograph the genitalia at all to compare that to Tabanidae between and the Hybomitra? Is there a difference at generic level?
No, because generally we don't catch the males, and the female genitalia is all very similar. So it's a general trend in Tabanidae that nobody does the male genitalia. As I say, unfortunately, we don't often catch the males.
Hi, thank you, Kirstin. So is this specific genus a vector for disease, or is it a problem in agriculture?
So they're biting flies. Because so little work has been done on them, we don't know if they are vectors of specific diseases yet. But if we catch more of them, we will then be able to screen for them. But it is a genus at the moment that has just had so little work done on it. That other specimen I showed the extra photograph of is definitely a Hybomitra, and we are going to hopefully go back to Monk's Kyle and try and collect some more material, because it seems like it's an area we don't expect to find tabanids, but we've actually found, we've probably got another species as well, new species that we've collected from the area. So yeah, we hopefully in the future would be able to tell what diseases they may spread.
So I have a question about those traps that you used.
Yes, the Manitoba traps.
Yeah. So I've read papers before that suggest that those traps work by, or they sort of use the polaritactic behavior of the flies and not temperature. So the flies see polarized light reflecting off of those balls, and then they're attracted to that.
There is an element to that, but there are different types of traps. So there are other traps called horizontal traps and glue traps, and they use black and blue material, and that's very much for the polarized light. So we do, for this particular group, we collected with these traps, but when we go out, we tend to put up all the different types of traps, so that we can catch them based on not just the smell and the heat, but also on the sight.
And like, which ones do you find the most effective?
We've actually just finished a 3-year project where we are going to compare the results to try and decide if any of them are better than the others. So we've been using 4 different trap types, and we're actually going to be looking at, we're in the process of doing the analysis for that.
Cool. I'm interested to see what comes of that. Yeah.
Any more questions? All right, if there aren't any more questions, thank you, Kirstin.
Sure.
So everyone, we'll be back after tea.